The science of keeping seeds alive has been the focus of many researchers for decades. We have a pretty good understanding of the factors that accelerate seed death. However, we still don't understand why seeds die. Hi everyone, I'm Nick and welcome to my lab. I have been fascinated by seeds for as long as I can remember. Seeds are this fascinating part of the plant life cycle that allows them to survive as the seasons change. Seeds develop in the canopies of most plants, and as seeds mature, they fall to the ground. On the ground, seeds must remain alive throughout the winter so that they can germinate and establish in the following spring. And it's this mechanism that we take advantage of in agriculture that allows us to extend seed longevity from a single season to many decades. Prior to modern refrigeration technology, farmers would set aside a portion of their crop to plant in the following spring. Farmers discovered early on that seeds must be kept cool and as dry as possible for them to survive throughout the winter. But it wasn't until we created gene banks that our understanding of seed longevity accelerated. Gene banks were established to save crop genetics that would otherwise be lost as new varieties were bred, varieties that yielded more, tasted better, or that were more pest resistant. The idea is that if we save crop genetics today, then breeders in the future can use those genetics to improve varieties in the future. For instance, crops today suffering from new and emerging pests can potentially benefit from crop genetics in the past. Today, gene banks are located throughout the world and are largely funded by governments. In the US, we have the National Plant Germplasm System, which is a network of regional plant gene banks that exist in different regions and focus on crops that are important to that region. For instance, the gene bank located in Geneva, New York focuses on vegetable crops and now cannabis, which is very important in that region. In total, there are 22 gene banks located throughout the US, with the largest located in Fort Collins, Colorado, called the National Laboratory for Genetic Resources Preservation, or NLGRP for short. The NLGRP was established in 1958 as a final backup for all crop species in the US. All seeds produced at regional gene banks in the US ultimately end up in Fort Collins, Colorado at NLGRP, and a small sample of those actually go to the Svalbard Seed Bank in Norway. Now, when the NLGRP was established in 1958, all seeds were stored at 5 degrees Celsius, which is like putting seeds in a refrigerator. But in 1977, the NLGRP and all other gene banks in the US lowered the temperature to minus 18 Celsius because new and emerging research demonstrated that colder was better. Now, there are many factors that reduce seed longevity. But the two most important factors that scientists agree on is temperature and seed water content. In general, seed longevity is extended as seeds are stored both colder and drier. The FAO, or the Food and Agriculture Organization, recommends that gene banks store seeds at minus 18 Celsius and 15% relative humidity, which is the global standard. Maximum seed longevity is achieved when seeds are stored both dry and cold. And the reason for this is how molecules behave in response to decreasing temperatures and drying conditions. For temperature, molecules slow down as the temperature is reduced, minimizing interactions and unwanted reactions from occurring. The current hypothesis is that as molecules interact with each other, they actually react chemically, changing bonds and ultimately degrading from what they originally were. And it's this degradation that actually reduces seed viability in storage. The same principle applies to seed water content, or how much moisture the seeds actually have when they're stored. If seeds are stored wet, then molecules can use that water to actually move around within cells, interacting, causing reactions, and ultimately degradation. Now, the reason why drying seeds works is sort of like putting a gate up around your house to keep your dog from running away. If you didn't have that gate, your dog would be able to roam freely. And the same thing is happening at the molecular level. As we dry seeds, Molecular movement is actually reduced, and the interactions between different molecules is actually reduced, which limits degradation over time. Now, over time, even when seeds are stored really dry and really cold, they still die, even when stored at really cold temperatures like liquid nitrogen conditions. And this is why old seeds don't germinate, or if they do, they grow really slowly and maybe look deformed. Over time in storage, it's the interaction between molecules that actually causes seed death. And unfortunately, we haven't quite figured out how to completely stop this from happening. So now that you have a basic understanding of why seeds die and the factors that accelerate seed death, I want to give you some tips for storing seeds at home. The first tip is to avoid storing wet seeds. Now, if you just planted your seeds for the season, there is the possibility that in handling the seeds, you actually cause those seeds to gain moisture, perhaps because your hands were wet, and in handling the seeds, you actually transferred some moisture to the seeds. What also causes seeds to gain moisture is if you're sowing them in a high humidity environment. So the best thing that you can do after handling your seeds is to bring them back inside into an air-conditioned environment so that they can dry out. I recommend storing your seeds inside in air conditioning for at least 1 day before you store them. This will allow your seeds to give up some of that moisture that they may have gained from handling or from the environment. The second tip is to use moisture-proof containers for storing your seeds. When you buy your seeds, they may have come in a Mylar bag, which is what gene banks use to store seeds for many decades. It's also what some seed companies will use to store their seeds. Mylar is the best material for storing seeds, but once you cut that bag open, those seeds are now exposed to the environment. Now, seed companies and gene banks will use a heat sealer to reseal those Mylar bags, but you most likely will not have a heat sealer at home. I don't have a heat sealer. And so the best thing that you can do is to store your seeds in a moisture-proof container. The best container for storing seeds at home is one that has a gasketed lid. Think of a food storage container or a glass jar with a gasketed lid. Those are your best options for storing seeds at home. If the container you choose doesn't have a gasket, then it's likely that the seeds within that container will gain moisture from the air and die relatively quickly. Now, my last suggestion is more of a warning. Moisture is the real enemy of seeds, and you may think that putting an open seed packet in the refrigerator is actually better than storing seeds at room temperature in a dry environment. But you also would be wrong. This is even a mistake that most commercial growers make. After sowing seeds for the season, they'll put their remaining seeds in a cooler without humidity control, and typically within a few years or even a season, those seeds will die. And that's because the relative humidity of most refrigerators or most coolers is actually pretty high. Storing seeds in that environment will actually accelerate viability loss. If you're gonna put seeds in a refrigerator or a freezer, make sure they're first put in a moisture-proof container before storing. I hope this video was interesting and it helps you understand the factors that accelerate viability loss. And I hope it helps you store your seeds better at home. See you on the next one.